Okay, we are gonna try to understand the big picture of the immune response. I try to make here a summary of how the immune system works. Of course, this is a very simplified version, so it doesn't pretend to be a very comprehensive explanation of how this actually works. Okay, here we have on the left side the area that represents the recognition of the threats, antigens. Can be bacteria, viruses, can be fungal infections, can be parasites, can be any foreign cell, can be any dead cell. Okay, we have in our tissues some cells that we call resident cells. Can be, for example, macrophages, can be dendritic cells. There are some pathogens that normally need to infect cells, so they are called intracellular pathogens, while others may be freely floating in the extracellular fluids. Okay, for example, we have here a dendritic cell that is in contact with the bacteria or virus and will phagocytose this bacteria, will break it down, and will process some of the proteins of this bacteria or virus and will place some of these proteins on its own cell surface. Okay, the dendritic cell will travel, okay, following the lymphatic vessels into a lymph node or any kind of lymphoid tissue nearby, and will present this antigen to the naive T cells. Okay, we have to clarify that this is the primary immune response. This is the first time we encounter this antigen, so it's going to take place a process that takes about 10 days or 2 weeks. This is the same thing that happens when, for example, we get a vaccine. Okay, the dendritic cell goes there, presents the antigen using the molecule MHC class II to the naive T cells. These cells are going to start dividing, a process known as clonal expansion. Some of these cells are going to become memory T cells. Other cells are going to become active T helper cells. Now, the active T helper cell will produce several types of responses, and the responses will depend on the type of antigen. It's not going to respond in the same way to extracellular, intracellular pathogens, or when there are parasites, or bacteria, or fungi, or viruses. Now, the active T helper cell will stimulate the macrophages to intensify the phagocytosis, and also will stimulate cytotoxic T cells, the CD8 cells, so they travel to the different tissues in which the infection is taking place and recognize cells that are infected by the same pathogen. Okay, it has to be the same pathogen that initiated this response. That's why we call this specific immune response. Now, the recognition of the cytotoxic T cells, of which cells contain the antigen, is made by the presentation of these antigens in the molecule MHC class I. So notice the interaction between nucleated cells and cytotoxic T cells, or CD8, is using the MHC class I, while the presentation of antigens to CD4 cells is by means of the the MHC class II molecule. Okay, this is what we call the cell-mediated immune response. Okay, this cytotoxic cell once recognizes the infected cell or the cancer cell or the damaged cell is going to destroy it. Now, what if the cells don't display the MHC class I? What if the virus messes up with the cell so it doesn't show up, doesn't show that it's infected? In that case, we have a very important cell of the innate immunity, that is the natural killer cell. The natural killer cell will destroy any cell that doesn't display MHC class I or displays very little of it. So they have more or less the same role, but one acts more specifically for cells that display MHC class I with a foreign protein, while natural killer cells will take care of every cell that doesn't have MHC class 1. I like sometimes to compare it to the highway patrol police. If they see a car that has any abnormality, doesn't have a license tag, doesn't have or has any damage, or is doing something weird, they are going to stop it, and they are going to, in some cases, arrest the person who is driving that car. Now, let's move to the other part here. What happens if the pathogens are not intracellular, if they are freely floating in the fluids? For that case, let's imagine there is an infection with viruses or some bacteria move following the lymphatic vessels into the lymph node. These freely floating pathogens or antigens are going to be detected by the naive B cells. Notice that they have receptors, which are antibodies on their surface, that are like antennas that recognize specific pathogens. So there is going to be an interaction with between the pathogen or antigen with the naive B cells. Naive B cells are going to do different things. They are going to start, first of all, dividing, producing this clonal expansion. That means from one cell, we develop thousands. Some of them are going to become memory cells. Other cells are going to become plasma cells, and we'll start making antibodies of the type IgM, which is the one that we produce during the acute infection, early during the infection. These IgM molecules will go out and will inundate our fluids and will bind to pathogens that are the ones that initiated all this. That's why this is also a type of specific adaptive immune response that we call humoral, because it's mediated by antibodies that act in the fluids. Humor means fluid, against any extracellular pathogen or antigen. Okay, these IgM antibodies are very effective since they have this shape. They are pentamers. They bind to many antigens. Now, the problem is that they don't bind very strongly. They don't have too much affinity for the antigen, so they might escape. That's the reason why we need the action of the T helper cell that will produce what we call the antibody class switching. Okay, at some point, with the help of these TH cells, the active T helper cells, the antibody class switching will occur, and the plasma cells will change from making IgM to making either IgG or IgA or IgE, which are antibodies that bind with more affinity strongly to these antigens. Okay, that occurs, and that signals that the person is protected, is immune to this specific pathogen. Now, let's take a look at this part here that we haven't mentioned. Okay, when the naive B cells get in contact with this antigen, they will engulf it. There is a process known as receptor-mediated endocytosis. There is going to be processing of this antigen, and there is going to be presentation of the antigen to the naive T cells in a similar way as it happens with the interaction between the dendritic cells and these naive T cells. The antigen is processed and presented using the MHC class 2 molecule, and the naive T cells are going to undergo the same process of clonal expansion, becoming active T helper cells, and will participate in the antibody class switching. Notice that the active T helper cell is also stimulating macrophages, so they perform the phagocytosis more strongly. So all of these pathogens, all of these antigens that are traveling, freely floating in the fluids, may be grabbed, okay, by these macrophages that will engulf them and destroy them here. Okay, this is in a nutshell what happens in the immune response. The only thing I have to mention is when we have enough antibodies in the circulation, okay, these proteins are going to make a negative feedback on the plasma cells, so we reduce the amount of cells that we are making. Okay, this is more complex. We haven't mentioned, for example, the different types of responses that the T helper cells may produce. There are different responses. The Th1 is the one that we see here. That is the one that activates macrophages enhances phagocytosis, but there is a Th2 response. In that case, we are gonna stimulate other types of cells, eosinophils. We are gonna stimulate the plasma cells to make IgE instead of IgG. Okay, that's why this type of response is involved in allergic reactions. Then we have the Th17, that if it's exaggerated, we are going to have certain types of autoimmune conditions. And we also have to mention the regulatory or suppressor T cell, that is the one that is going to keep all of the other cells under control, so the immune response is not very exaggerated. Okay, in a future encounter, we could talk more about these different types of T helper cell responses. Okay, hope you understood, hope you liked it, and please contact me with any comments, any questions. I'll be more than glad of helping and learning also with these comments and these recommendations. Thank you very much.